Well, especially now that I'm retired, I get to do only the things I like to do, which is the insect work. So we always had some kind of a new project that could challenge me, because I started really, my expectation was to go to graduate school, but I'd finished my undergraduate and I wanted to take a little break, and so I asked, and I had just taken Robin Vannote's course, and he had just gotten the River Continuum grant. And I said, "You got any jobs?" And he said, "Yeah." And so we started on that, and I just had so much fun doing the stuff. Each new project that came that I never got around to doing graduate school. We've always got something fresh going, whether it's temperature-related things or toxicity things, but I always manage to keep somewhat of a taxonomic focus on things, which was always my first interest, is figuring out who's who. There probably wasn't one moment, because that project was a culmination of about 30 years of stuff with some big breaks in the interim. But it started because we had Amylitis mayflies in White Clay Creek here that we knew a whole lot about their biology, their life history and all that. But we knew also that they were parthenic and they were all females. And the taxonomy of that group has always been based on adult male genitalia, and we don't have that with these. And it was always a question of really, well, what do we call a species? Because we have, by the books, we had two different ones out here. They looked slightly different in their color, but they did exactly the same thing at the same time. This means, in this case, all the individuals in the population are females. They don't need to mate to reproduce, so they all produce eggs that hatch, and they're genetically identical to their mothers. They're clonal in that way, and in fact, in the case of these mayflies, there's no going back because they're also triploid, so they can't even do meiosis, which is what you need to do to create, the eggs that can normally be fertilized by sperm. So partgenesis is actually really, it's quite widespread in mayflies. We have 50 mayfly species in White Clay Creek here, and 7 or 8 of them are exclusively female parthenic. But some of those are ones that are parthenic populations of ones that are sexual elsewhere, and they're not triploid, they're diploid like normal things, but they're still clonal. But for some reason in mayflies, it's really common. Parthgenesis is really common. What's unusual with these Amylitis ones is this is the only case in mayflies that's ever been documented of polyploidy, the triploid things, and a clear hybrid origin. We know the parents were. So that's what's unusual in this case. I started this project to try to figure out, like, where did these come from? Because parthenic species their ancestors were sexual. So we know that because the parthenogens, even though they have some short-term success, in terms of evolutionary success, they're always at the tips of evolutionary trees. So we don't see speciation once they've gone parthenic. It seems to be a dead end, and they don't last a long time, but they can be really successful and abundant. And in the case of these, probably 99.9% of every Amylitis that's ever collected in eastern North America is one of these parthogens. In the case of many parthogens, they don't swarm anymore. But it's interesting because the Amylitis are different that way. So they apparently have retained that swarming behavior, these females, they're like that. they don't mate, but they seem to need to go through the motions. And the way I know that is because all the other parthenic mayflies I work with here, and I worked with a lot of them, because we tend to use them for experiments and things because they're easy to culture in the laboratory. So, because they don't need to mate. Because mating in mayflies generally occurs in conditions that you can't really replicate, like, outside over a tree at a certain time. So the other parthenic mayflies that I work with in the lab, once they become an adult, if I just drop them on water, they'll dump their eggs. But the Amylitis won't do that. They're kind of a pain that way for me, because they won't. And the only time I've gotten them to dump their eggs on their own is when I've been outside and I see one coming down out of like an imaginary swarm. And then if I grab that female on the wing and put her on the water, she will lay her eggs. So they seem to have retained that behavior, even though they don't need to mate to complete things, but they do need to go through the motions. So they're overwhelmingly abundant compared to all their ancestors. But I wanted to get to the bottom of where they came from, and so it was a hunt that took a lot of years to find the relic sexual populations that had given rise to them. If there was an aha moment, it was once we had some of the more modern genetic techniques where we could say, oh, this one was your mother, and therefore this one was your father. And so it turns out that these parthogens are hybrid origin. They happened when probably late Pleistocene, Ice Age, when the ice sheets came down North America, there were two sexual species that were geographically isolated, but the glacier essentially forced the northern one down into where the southern one's range, and that's when these hybridizations occurred. And these hybridizations gave rise instantly to these parthenic lines that were not only a hybrid, but they were triploid, which meant they had 3 sets of chromosomes, and they became parthenic and clonal. And so this happened probably a number of times, but only in a really local spot, and created a bunch of different genotypes that then went crazy and colonized the whole eastern half of the country, and probably displaced most of their sexual ancestors. That's why it took 30 years to find them, right? And there's only tiny little relic populations of the sexuals. And where we find the sexuals, there aren't any of the parthogens there at that spot. I started doing genetics on them back in the 80s with looking at nuclear markers. But then we started with barcoding, which is a mitochondrial gene, in the 2000s. And the advantage of mitochondrial genes is you can sequence things and look very finely at things, but the disadvantage in most cases is that you're only looking at a matrilineage. You only know who the mother's side of the gene tree. But in this case, that was the way we got the answer as to who was who in this formational event. And so these two species we have out here had different fathers but the same mother. Discovery of the new species was kind of incidental to this, because 4 of those new species are ones that were involved in these hybrid events that created the parthogens. It's not terribly unusual to find new species even in North America of things, especially when you can look at them at a level that we didn't used to be able to look at them with using some of the genetic techniques. And things are much more diverse than anybody really acknowledges.